Hello class, I'm Teresa.
I'm Jordan.
And I'm Habib. Today we'll be your guides in tackling the technique of qPCR.
What is qPCR? Real-time PCR, or quantitative real-time PCR, is a means by which PCR products generated during the course of the PCR reaction are detected and quantitated in real time. This process uses a fluorescent dye to measure products as they are produced at each step along the course of the reaction. Thus, this method eliminates the need for gel electrophoresis. When did this process come about?
In 1971, Kjell-Kleppe and Gobine-Corona first published a description of a process that sounded similar to PCR. However, it could not be verified as PCR because primer technology required for this process was not readily available at that time. Then, in 1985, Kary Mullis invented the process that we now call PCR. He was then awarded the Nobel Prize for this invention in 1993. In that same year, Higuchi and his colleagues recognized that the process of PCR could be modified by utilizing fluorescence to monitor the progress of the reaction. This would now become known as qPCR.
Before looking at the process of qPCR, let's recall the process and components of regular PCR, as qPCR builds off this common technique. PCR is a technique used to amplify a specific gene or portion of DNA. PCR utilizes a series of reagents and a cycling machine to increase the number of copies of your desired gene. To properly conduct PCR, a buffer, which contains proper salt concentration and pH for the reaction to take place, dNTPs, which are extra bases to be incorporated to the new copies of DNA, primers, which are short sequences of DNA that are complementary to the gene of interest, a Taq polymerase, which is a polymerase enzyme that works well at high temperatures to transcribe new copies of DNA, and water to bring the reaction to the correct volume, are combined in a reaction vessel. The reaction is then placed in a PCR cycling machine that goes through a series of denaturing, annealing, and extension steps in order to effectively replicate your desired gene of interest.
During the denaturing process, the temperature is raised to 94 degrees Celsius, which allows the double-stranded DNA to separate. During the annealing process, the primers bind to the DNA at a temperature that is primer-specific. During the extension process, the polymerase binds to the primers and adds complementary bases, making a new copy of your desired gene. The extension time is product-specific, meaning that 30 seconds corresponds to about a 500 base pair product. This process is completed in a cycling process to eventually obtain your target sequence. As you can see, after completing about 3 to 4 cycles, you already have your desired product. qPCR utilizes this same process, but incorporates fluorescence in order to effectively quantitate the amount of product that would result.
How exactly is qPCR different from the process just described? qPCR is different from regular PCR in that it utilizes fluorescent dyes to quantify the amount of product that is obtained after each cycle by the real-time PCR machine.
How is fluorescence in qPCR quantified, though? There are 2 different ways in which fluorescence is incorporated into the amplicon and then quantified. The first method uses an intercalated dye to directly incorporate the fluorescence into any double strand of DNA, meaning that it is a nonspecific detection method. Cyber green dye is most commonly used. This method is ideal when a single amplicon is subjected to analysis. The second detection method uses primers specific to the gene of interest. This method is more common and is what we have depicted here in the animation. These primers are labeled with a fluorescent dye and a quencher. Depending on the type of primer used, it will only fluoresce when annealed to the template, such as a molecular beacons, or when the dye is removed or cut from the primer during the extension, such as with Taqman probes. The qPCR machine is then able to record and quantify the fluorescence that occurs at each cycle of the replication process. The amount of fluorescence becomes measurable after a threshold cycle or crossing point has been reached. This threshold value will now be discussed next.
What results are expected for qPCR? Once the endpoint is reached, the number of copies of DNA is analyzed and compared with the fluorescence of a known standard in order to generate a comparison curve to identify amount of cDNA in an unknown sample. All samples are compared to the standard or housekeeping gene, as it's considered a stable value for gene expression analysis. The number of DNA copies in a sample can be analyzed once the threshold value is reached, which can be useful in diagnostic tests, for example when trying to detect the presence of metastatic cancer genes in tissue or cell samples. In other words, you can be sure of the presence of a specific gene in the sample after the threshold is reached. Verification of the presence of your specific gene can be done without having to run the results through gel electrophoresis. In regular PCR, you have to run the PCR reactions out on a gel in order to confirm the presence or absence of your gene of interest. When fluorescent markers are utilized, the final step of running samples out on a gel can be eliminated.
Now, let's put everything together. First, when conducting qPCR, a qPCR reaction is prepared with the addition of fluorescent dyes or fluorescently labeled primers. Then, the reaction is placed in the qPCR machine. Fluorescence will then be produced during the reaction and measured by the machine in real time. The amount of product can then instantly be compared to a known standard, which is the number of copies produced at each cycle in the process. And that's qPCR in a nutshell. Or should I say, banana peel.
What is qPCR used for? qPCR is generally used for gene expression analysis. This analysis gives us insights as to how regulation of mRNA expression is part of the complex process of cell control. Gene expression analysis also has real-world applications. For instance, it can aid in diagnosing patients with pathogenic infections by detecting the presence of a pathogen-specific gene once the fluorescence value threshold has been reached. Recently, qPCR has also found uses in detecting plant pathogens. Molecular methods are increasingly being used to detect, quantify, and study microbial populations in food or during food processes. Among these methods, PCR-based techniques have been the subject of considerable focus, and ISO guidelines have been established for the detection of foodborne pathogens. qPCR is considered as a method of choice for the detection and quantification of microorganisms. One of its major advantages is its faster than conventional culture-based methods. It is also highly sensitive and enables simultaneous detection of different microorganisms. What are the advantages of using qPCR? This technique has improved precision in the quantification of target sequences. It is capable of measuring initial concentration of target DNA over a vast dynamic range and with a high degree of sensitivity. As little as one copy of template DNA is initially needed. It allows for the ability to process many samples at the same time. This technique increases the overall speed of gene expression analysis. There's no need to complete full amounts of cycles if simply looking for gene presence once fluorescence detection is available. Thus, you are able to quickly confirm diagnosis. qPCR also allows you to collect data during the reaction process, so there's no need to run a gel after the endpoint is reached. Thus, you are able to instantly confirm presence or lack of presence of intended gene samples.
But what are the disadvantages of qPCR? Researchers often face challenges in reliability and reproducibility because of the lack of assay standardization. Therefore, it is very important to optimize the reagents and reaction components, including proper internal and external controls, and perform rigorous data analyses in order to generate accurate and reproducible results in qPCR experiments. The standard housekeeping genes for every type of analysis may also not be readily available, so optimization of proper housekeeping genes needs to be completed prior to any experimentation. Additionally, qPCR is typically more expensive to conduct than regular PCR due to the special cyber green and TACMAN dyes. When conducting qPCR, we often face variability in template challenges, as mRNA expression is context-dependent or highly variable between samples. This technique expresses the importance of obtaining viable samples without wasting any, for each sample can give a unique analysis.
Now let's talk about some recent advancements that they've been doing for this technique. Better pre-amplification methods are being utilized in order to standardize the samples. This allows for the profiling of large numbers of markers, starting with minute sample amounts such as single cells, and determining the quantity of cDNA in the sample. An approach known as selfie qPCR was recently created in which no reference gene or housekeeping gene is required. Instead, a comparable absolute digital expression level will be generated to share between all of the labs, thus eliminating the need for multiple reference genes. Another new development is digital PCR, in which a sample is diluted and divided into hundreds of reaction chambers, so that each chamber has about one copy of template to begin. This method is useful for quantification of rare mutations among a high population of wild-type sequences.
As your guides to conducting qPCR, we hope you all enjoyed the introduction to this very useful technique in molecular biology experiments. Now, go out and explore the world of qPCR.